what I'm going to talk about is how can vaccines mitigate AMR, and this is going to be two parts. One is on the antimicrobial use and AMR itself. I mean, it's almost like textbook knowledge, and everybody says because of the use, underuse, misuse, whatever you want to call, we have AMR. So, is there evidence? Beyond doing an experiment in the laboratory where you use E. coli and small amounts of ampicillin, and then finding out after 12 hours that this E. coli has grown resistant, do we have real life evidence to show that antimicrobial use actually leads to AMR? That's what I want to cover in the first half, and then in the short, few slides, I'll give a couple of main examples of how vaccines have played a role in mitigating AMR. So, acknowledgements. My foray into microbiology started with this fellow. He is no more. He is Professor Trilochan Makkar. He was at Curtin University in Australia, Perth, Australia. For some reason, he wanted me to work on a project on Staphylococcus aureus associated with mastitis. And up until then, I had been a virologist. So I asked him, I'm a virologist, why do you want me? And he says, I don't care, I want you. It doesn't matter what the organism, but I want you. I said, okay, fine, let me learn with you. So with him came Dr. Islo, because I couldn't have got into mastitis or Staph aureus without having a friend of mine who had already been working on some of these aspects. And then came a few people into my lab who started working on some of these projects. This is Dr. Raju Sunadar. He is now in Indian Immunologicals. This is Deepak Rahul. He is now a principal scientist in National Meat Research Institute. And this is Nicola Williams, with whom we had some projects with looking at AMR in broiler poultry production, meat production. And of course, this is the team we had with UK that they visited at one of the poultry farms. So I could not have gone into this field without these people. And of course, there's Dr. Harsha Kumar Shetty. He belongs to Venkateshwara Hatcheries, who facilitated some of our AMR work in poultry. So, just a little bit background. Where do we stand and what are they? Bacteria are everywhere. 60,000 different species in the soil, billions per gram, 10 to the 6 per ml of fresh water, wide variety on our body, 150 species on our hands. Healthy hands can carry up to 1,500 colony forming units per centimeter square. And for healthcare workers, it can go as much as 40,000 to 5 million. So they are there everywhere in our body. The original hypothesis was that there were 3 bacteria to 1 nucleated cells, but this has now been recalibrated. The new numbers are 1:1. So as many number of cells as we have in our body, we have that many number of bacteria. And they actually form 0.3% of our body mass. This was also recalibrated. 210 grams, if you weigh 70 kgs, 210 grams of your body is bacteria. So we coexist. We have to coexist. They help us and we have to help them survive. But they are also major killers in developing countries. And here is actually an old data from 2009. This is the pneumococcal deaths in children under 5 years. And this is actually number of children under 5 years. So you can see India has a huge number of, or had a huge number of children, but not much death. But if you go to some of the underprivileged countries like Afghanistan, Angola, Democratic Republic of Congo, numbers are low, but the deaths are high. And antimicrobials probably have played a critical role in restricting that. Sanitation is one, but antimicrobials have played a critical role. This graph shows starting from the use of penicillin. So this was for sulfur drugs in the 1930s, then the penicillin came in the 1940s, and then basically drastic reduction in the US of infection death rate per 1 lakh population. So you can see very low after the antibiotics were discovered. So stopping antibiotics is not a solution. Believe me, we cannot live without antibiotics. We have to coexist with antibiotics, but we have to use them rationally. The bugs also are in warfare. They don't want to just die, so they will also develop resistance. And the first instance of how these bugs develop resistance goes to Stanley Falco. His PhD thesis was on an episomic element in a strain of Salmonella typhosa in 1961 from Brown University. So he was the first one to actually show genetic transmission of antibiotic resistance. And how does it all appear? If you can see in this graph, so this is 1930, within 10 years resistance to sulfonamide was detected. Penicillin, less than 10 years. The longest was probably vancomycin, which is also not even 20 years. Within a very short span, people have identified resistance. Why is it? Okay, so remember, no new classes, this has been beaten to death now since the 80s. Very few antibiotics have been discovered. But what we need to remember is that this resistance that is discovered is the date of discovery, not the date of existence. It may have existed before, but people were not looking until an antibiotic was discovered. Okay? So, these are just the dates of discovery, not existence. So, here's the chicken and egg question. Did the resistance exist before? Or did we cause the resistance? What came first? Okay, resistance or so somebody has done some experiments and this has been published. So they took out permafrost samples around 30,000 years ago and they simply did some metagenomics. And what they found? Resistance to glycopeptides, resistance to tetracycline. That means there were bacteria there that already had these genes. So it's not that resistance has appeared now. Resistance existed before. It's just that we have amplified it by misuse, overuse, whatever you want to call. So much so that this now we call is a ticking time bomb. This I came across, this is actually a diagram from a high school student in Singapore. So he calls antibiotics are a double-edged sword. And he sees something called as a certain unseeability. So you can use antibiotics, but you can't see after which time you will see resistance and you have problems. So in his language he says, curtain of unseeability. This is a simple diagram for a high school student, but this is very true. Okay? So, and well, there is no reason to panic. I think it's only prudent that we make preparations to panic. And this is exactly what we are doing. Stewardship or whatever else that we want to do. Okay? So what are the reasons? And I will only point out the two major ones that are here. So on this axis is evidence that the factor is contributing to AMR, and on this axis is the relative contribution of the factors. Okay, so here the big blocks here on this side are human antimicrobial misuse and overuse, animal antimicrobial misuse and overuse. And believe it or not, both of them are because of us. It's all anthropocentric. We want food animals because we want our food security and we want to be healthy. It's all human driven. So I thought this is KSTA, I'll put at least one Kannada sentence. So those of you who can read it. But then as I said, what is the field level evidence? Does AMU correlate with AMR? This graph shows penicillin resistance. So this is the penicillin resistance on y-axis, and this is the total antibiotic dose, what is known as the defined daily dose per 1,000 population in various countries. This is of course in Europe, but you can see a linear relationship. But there is a hitch. This is only an association. This does not say about a cause and effect. It does not show that because of you're using, you're seeing an AMR. It just shows that there is an association. Okay? This is also being shown in other countries which are not the so-called first world countries. These are not the first world countries. So again a linear relationship. And recently, very recently, this has come out in 29 to 2021 global burden of disease data. So here you can see prevalence of ESBL and or AMP-C in food producing animals, and this is the consumption of cephalosporins in food producing animals. And you can see the increase, and pretty much, there is a slight curve, but it shows that there is an association between use and existence of resistance. And I've put up this thing. It's an interesting graph, very difficult to read, but you have to really, focus with a microscope. This actually is from 2001 to 2010, percentage resistance of various organisms in organic farm. These are livestock farms, and this is conventional farms which use antibiotics. Okay. Really, if you compare, you don't see much of a difference, but in some years you see a huge difference. In 2001 you can see the difference, and in some cases if you actually draw a line, the line doesn't look that different. But believe me, if you look at the average and the standard errors, you will see difference in that organic farms have a much lesser percentage of resistance. And this is also shown here, specific populations, here cattle, and if you see, this is about, I don't know, 0.12 or something. This is slightly more, so 0.15 or 0.2 or something. So obviously in conventional farms which use antibiotics, you see higher resistance. So what do we do? What do we do about this AMR? Of course, we have been talking about these alternatives. So there are various alternatives, including bacteriophages, probiotics, prebiotics, maybe newer drugs, phytochemicals, etc. And I am going to talk in the next few slides, try to impress upon you how vaccines can combat AMR. This sort of, looks counterproductive. Why would a vaccine be useful in reducing AMR? Okay, so this is actually the use of vaccines, and this shows the life expectancy in years. Of course, most of these are viral vaccines. Of course, there is hygiene, clean water, rabies vaccine, antibiotics as well. So you can see the increase in life expectancy over the years since the time we have introduced vaccines. And how does it actually work? In a population, if you look, if there is a single individual and there is an infection, and he uses the first line of antibiotics. If the infection is not cleared owing to AMR, a second line antibiotic is used, and he or she may become resistant, or infection could be cleared. Okay. If the person is resistant in a population, that person can spread the bugs to others, and the other people can get these resistant organisms. If you apply a vaccine at an individual level, if the individual is already vaccinated and the bug comes in, individual clears. There is no resistance. Okay. At the population level, because this individual is already, several people are already vaccinated, you can imagine everybody who is in and around this person is protected. The person who is not, who is on the fringes, is the one who gets infected. So this is the population level effect of a vaccine against resistant bugs. Okay. And I also wanted to put this, you have already seen this, the development of resistance to various antibiotics, but this is actually the development of resistance to vaccines. Okay. One of them is eradicated smallpox, and there are bacterial vaccines, tetanus, pertussis, polio, others, and you will see a couple of them where you see resistance, and this resistance is not because the bugs themselves have become resistant to the vaccine. It is because the original strains that were used against were particular serotypes, and the serotypes have changed. So you see the disease because you don't have the serotype in the vaccine. And I'll give some examples of that. So what do we target in terms of AMR? Of course, AMR traditionally has been talked about by antibiotics, antimicrobials. Less people talk about antivirals, antifungals, but they are also antimicrobials. So what we target is typically the escape pathogens, standard pathogens which is defined by WHO and everybody else. So is there evidence? So I've showed you a diagram, population level, herd immunity and all that. Do we really have field level evidence that there is reduction in AMR because we use vaccines? Okay, I'll take two examples. One is strep pneumoniae. This is a common respiratory tract pathogen of humans. In fact, up to 90% of us carry them in our nose. Different serotypes, different variants. Okay, there are more than 100 serotypes of strep pneumoniae, and each of us could be carrying different serotypes. They cause pneumonia, bacteremia, meningitis, acute otitis media, septicemia, all different kinds of diseases. And the other one is Haemophilus influenzae. For both of these, we have traditional vaccines available for several years. So that is why we have much more data on what happens when you use vaccines for these. This also causes similar infections, respiratory tract infections, similar to these, but few more extended diseases. This is one example of what happens if you use strep pneumonia vaccines. So these are called pneumococcal vaccines, and these are examples of invasive pneumococcal disease, basically bloodstream infections. So there are, as I said, 100 serotypes are there. The first vaccine only contained 7 serotypes, and then 13 serotypes. Now we have 23. So this is a multivariant vaccine. So when PCV7 was introduced, if you look at PCV7, reduction from the time PCV7 was introduced, but if you look at other strains are still existing, and these were contained by PCV13, and then there are other strains that are not even PCV3, PCV13 or PCV7 that are still there, but those are not yet resistant. Maybe they will gain resistance, and how many valents we want to make, I have no idea. So this is Haemophilus influenzae, and you can see the diagram here also. As soon as the Hib vaccine was introduced, precipitous drop in the number of cases of Haemophilus. But if you see the ones that are non-typable, which are not Haemophilus influenzae B, there is a slow increase. Okay, number of cases of Haemophilus influenzae. So when multiple serotypes are involved, you always have this problem of other serotypes overtaking and causing the same disease. The question is, I talked about disease. Is there a reduction in AMR? So this one simple graph is talking about IPD cases per, so this is invasive pneumococcal disease per 100,000 over the several years, and the introduction of PCV13, and this is basically the reduction in various antibiotic resistance, 63% to macrolides, 81% to cephalosporins and tetracyclines, 83% to penicillins. This is actually real world data showing that vaccines do indeed reduce the incidence of AMR. Okay, how does it work? I've already given a couple of diagrams. So basically at the population level you get decreased infections, safeguard the communities, decreased antibiotic use, reduce the resistance evolution and spread, and then individual as well as individual levels are protected, and you also preserve the antibiotics. You don't have to go for higher level antibiotics. So the burden of AMR, which can be either private or public, and in the case of private you can have individual or household or individual livestock, and at public level you can have health systems including human and animals or the ecosystem. And basically you look at the cost of illness, cost of prevention, cost of intangibles and opportunity cost, and these include leaves you have to take for your children because the child is sick, he or she cannot go to school, you have to stay at home to take care of the child. All these are also included in the intangible costs. So all these can actually be factored in to show that vaccines indeed contribute to reduction of AMR and the ramifications. Okay, so Lipsitch and Sieber actually generated some information. Let's say you vaccinate against an agent A, you get either no or poor infection, most probably clearance. So either existent or treatment driven resistance can't be worse. You can't get worse than what is already existing if you have already vaccinated. Or you don't have to use antimicrobials at all. When you don't use antimicrobial antibiotics or antimicrobials, you don't get resistance. So less opportunity for resistance selection and reduced bystander resistance selection in normal flora. Remember, sometimes you also get normal flora that become resistant. As I said, 100 serotypes of strep pneumonia, some of them are just hanging around. You may not have a disease due to that particular strain, but because you used an antibiotic, they may gain an upper hand. So that is also reduced. Reduced transmission from of agent A to contacts which are either vaccinated or not. This is the herd effect, and of course again reduced bystander effect. And you can have reduced need for broad spectrum or empirical treatment to cover the variants that may resist narrow spectrum treatment. Now you can actually use probably penicillin instead of using 4th generation cephalosporin. So if the vaccine is not colonized with agent A, you can still have reduced transmission. or reduce population of agent A to exchange resistance elements. So if you assume that 100 bacteria will invade you and you are already vaccinated, only 10 are able to invade because of the immune system, then you are actually reducing the numbers. And you can have preferential effect versus the resistant strains of A, and you will have reduced selection for resistance. So there is a whole lot of things that go on by use of vaccines on the effect of AMR. So question is, what is the impact? What really is the impact? How can we see this in the real world? That is a problem. If you don't tell this to WHO or policymakers, they will not believe. They want numbers. They want to see, okay, 80 crore rupees saved. By using a vaccine, I will save 80 crore rupees. How do I arrive at this 80 crore rupees? And these are very difficult things to actually measure, especially because the effect on AMR or the consequences of AMR itself is a secondary measurement. And now you are putting a tertiary measurement overlapping the vaccine on it. So if you look at a scenario here, if you actually take a pre-vaccine AMR burden at the childhood stage, you have high, and then you can actually reduce by the post-vaccine burden at the childhood stage, and even as an adult, you might actually reduce the numbers. So this is actually beneficial. And in fact, these are taken from WHO. They have done some statistics as to what exactly happens. So this is looking at vaccine avertable, basically how much can we prevent AMR burden by infectious syndrome and pathogen. So the syndromes here are bloodstream infections, tuberculosis, lower respiratory tract and thorax infections, and the others. So this is basically the percentage of infections that you can reduce by using vaccines. And vaccine averted deaths. So this is different vaccines that are given here, including TB, Klebsiella pneumoniae, Streptococcus, Staph aureus, various things. So you can see 1 lakh deaths could be averted by using TB vaccine. Similarly, if you look at vaccine averted disability adjusted life years, again, Strep pneumoniae, 5 million, and then goes down. So this data is now available. So policymakers can now see what is the vaccine doing and how much can be saved. This is basically dailies, and this is look at infectious syndromes. If you use vaccines against lower respiratory tract infections, tuberculosis, bloodstream infections, what is the vaccine avertable deaths, and similarly what is the dailies that you can save with various things, and in this case 10 million. So if you have vaccines against lower respiratory tract infections, you can actually save 10 million. Okay, so these are vaccine averted hospital costs. Again, remember the costs are manifold. You can have deaths, hospital costs, community costs, other spending. So this is in US dollars. Again, different kind of vaccines. This is in billions. $8 billion US can be saved by a hospital cost can be saved by one Staph aureus vaccine alone. Vaccine averted productivity losses. Again, as I said, child care or some farmer who is unable to go to the farm. These are productivity losses in US dollars. Again, Strep pneumoniae, $3,500 million, and goes on and on. So WHO has a site where you can actually go and pick out the organism of your pet organism, if I can say so. You can go and check what can it do if you have a vaccine. So I'll come a little step backwards. I've talked about bacteria, AMR, bacterial vaccines. Do viral vaccines have a role in AMR? Remember, I'm talking mostly about bacterial resistance, not about viral resistance. If I use a viral vaccine, and famous example would be influenza. If I use influenza vaccine, can I avert AMR and the costs associated? Of course yes, because in many cases, viruses are the primary pathogens that cause the problem. And then bacterial infections are secondary. But then now how do we put in all these calculations and costs? This becomes a little complex. Okay, so this diagram sort of gives you some idea of what happens. On this side is the intensity and this is the time. and the blue one here is the viral titer bump as bacterial infection is established. So here is the secondary bacterial infection, and then there is impairment of bacterial clearance, which leads to overwhelming infection. Okay, so this is basically, in the number of days, it's 5 to 7 days for the virus to replicate, and then the virus probably goes away, but then the bacteria causes the secondary infection. And Dr. Islur was talking about animals, so I thought I'll put one diagram on animal infection. So these are basically, these are viruses on this side. These are vaccines against respiratory diseases, and then also looking at co-infections with these. So the impact of sequential co-infections of respiratory pathology in cattle. So if you look at Pasteurella hemolytica and BRSV, this is redder than BHV1, for example. That shows that overlapping with another bacterial infection increases the severity of disease. So this severity could be reduced simply by using a viral vaccine. Okay. So the consequences of viral vaccines, examples I'm taking acute febrile illness or respiratory tract infections. If you diagnose, still you have fear of secondary bacterial infections. If somebody diagnoses if this is influenza, you're not supposed to give antibiotics. But the doctor will prescribe antibiotics anyway, because he or she is worried, I don't know what will happen, you better be safe. I'll prescribe something anyway. So that is one thing. And the other way is if there is no diagnosis. This happens in most of the cases. You have a runny nose, you don't know what it is. You have some wheeziness and things like that. So most of the time it's not diagnosed, and the doctor directly goes for antibiotic prescription. Or if there is no antimicrobial prescription, you have less opportunity for resistance. This is actually the problem in viral infections. So by using vaccines against viruses, you could actually reduce AMR. So consequences of respiratory tract infections: socioeconomic impact, frequent medical visits, healthcare costs, work or school absenteeism, deteriorated lung infections, secondary infections, wheezing, asthma, antibiotic collateral damage, bacterial resistance and microbiota impairment, and decreased quality of life. So when somebody wants to go to a policymaker, all these needs to be hammered, and you need to tell what are these, what are the savings when you use a vaccine. One example I just want to highlight: how does COVID-19 contribute to AMR? Antibiotic therapy for COVID-19 patients were as high as 72% in some hospitals. Only 8% of the COVID-19 patients were found to have bacterial or fungal co-infections. I'm just throwing it out. This is data available. So any cold, you will first see that prescription of antibiotics. Even some of us go to the pharmacy and, can you give me amoxicillin? We are also responsible for this. Okay. So in summary, at the individual level, you see prevention of infection, inhibition of colonization of bacteria, limiting spread within the body, and then because of the vaccine you see a memory response, so you get an extended period of protection. With antibiotics it's only very short time. As long as the T-half is there in your body, you can be assured that it's working. After the T-half is over, done. Maybe 5 days, 7 days. If some other infection comes, you have to take it again. Vaccine is not like that. Minimize the effect on the microbiome, because antibiotics also affect our microbiome. At the hospital level, minimizing the OPD visits, reducing the spread within the hospital. At the community level, curb the pathogens when at a low level. Imagine you have an infection and you have to give an antibiotic. At the time you have an infection and you are diagnosed, you are loaded with bacteria, and that's when you are giving antibiotics. But vaccines are given when you may not even have the pathogen, or you may have very low levels of the pathogen where you don't see the disease. So that is a major difference between the two. And of course you see decreased transmission and herd immunity, which is also protecting people at risk, for example elderly people or people with immunosuppression. Overall, there is minimization of treatment costs, reducing prescription of the antimicrobials, prescription and use of the antimicrobials, and then facilitating the rational use. Sometimes you can actually go to the narrow spectrum. But then vaccines are not alone sufficient. As we have been talking since yesterday, and we will go into tomorrow as well, it has to be a multi-pronged approach. Okay, we do all this science, but Science is just the foundation. If you want to really take it to the masses, somebody else needs to step in. Dr. Gafoor was here this morning talking about inventions and going into marketing and things like that. We need that as well. And ultimately, we have to use a program where everything is in place. Just not having, even if we have all these strategies available scientifically, is not sufficient. So we need to have everything in place, including